Ya está.
So, welcome everybody to this session entitled Neuroimaging and Neurotechnology. And as we are on time, we are going to start right now. The first presenter is also a co-chair of this session, is Gemma Vera. So, Gemma, when you want.
Hello, everyone, and welcome. My talk is titled Automated Two-Photon Targeted Multi-Patch Clamping Robot. I know this might seem like a mouthful, so we're going to slowly explain what we're trying to do. But the idea is that we're trying to automate a technique called patch clamp. So what is patch clamp? So patch clamp is the gold standard technique to determine cell activity, function, and type. So it's mainly done in cells that have a electrical activity, so think neurons, cardiomyocytes, some cells in the GI tract, etc. Now, this technique, we're doing it in neurons, in mice, but as I said, can be applied to many other cells. And even though it's a very basic, very needed technique, it has a very steep learning curve. It requires a lot of skill, and even when you're very skilled, it has a very low throughput. So obviously, this is a technique that could really use automation, not only to increase the throughput, but also to avoid this very steep learning curve. So physically, what does it mean to do patch clamp? So you're going to have a pipette that is going to be filled with a liquid that resembles the inside of the cell. And with the use of our manipulator, you're going to put the pipette slowly, approach the cell. And then when it's in contact, you're going to apply subtle suction. What this is going to achieve is that you're going to have the cell membrane be sucked into the pipette. And this is going to be called a gigaseal because it's basically a seal that is in the giga-ohm realm or range. And then you're going to apply further suction, again, subtle. And what you're going to do then is basically you're going to be breaking the membrane of the cell, and then you're going to have a continuum between the inside of the pipette and the inside of the cell. And because you're going to have an electrode inside the pipette, and you're going to also have a bath electrode, then now it means that we can both stimulate and record from the cell. Now, when we do multi-patch clamp, it just basically means that we're going to do patch clamp in several cells at the same time. So what is the use of that? Well, first of all, you're going to be able to see how different cells are connected, and you're going to be able to detect even subthreshold correlations at a very high temporal accuracy. So this is something that cannot be done with any other technique, not even calcium imaging like GCAM, for example. That's something that this kind of accuracy cannot be managed with any other technique. So, for example, we have here these two neurons that I patched. And as you may know, neurons communicate to each other through electrical impulses, and they have synapses to transmit these signals. And so the cell on the left here, called pre because it's presynaptic, that cell was synapsing onto the cell on the right, the postsynaptic cell. And so basically, if we stimulate the presynaptic cell here in green, and we make it spike 5 different times, then we can actually see the EPSPs on the postsynaptic cell. So that just means that we're basically exciting, the second cell is seeing the excitation that the first one is receiving. And obviously, if we do it with more cells at the same time, then we can start seeing what kind of relationships are between the cells. So for example, these 4 cells here, that I patched, I then later, by stimulating each of them, I could see that actually 1 and 4 were connected to each other reciprocally. So that means 4 was synapsing onto 1 and 1 onto 4. And then 1 also was synapsing onto 3. So stimulation of 1 would be felt by both 3 and 4. And not only this, but if you stimulate the cell number 1 at a high enough frequency, we could actually see some inhibition on cell number 2. And this is actually because there was an interneuron, which is an inhibitory neuron, that basically was being excited by cell number 1 and then in turn inhibiting cell number 2. So as you can see by multi-patching, you can get a lot of information on how cells connect to each other, how cells talk to each other, and not even only the 4 cells that you're patching, but also even other cells in the neighborhood. So it gives a picture of the microcircuits in the brain. Now, when I talk about two-photon targeted, it just basically means that we want to know what type of cells we're going to be patching. So if we are able to express a fluorescent protein in the cells that we want to patch, for example, EGFP, then we're going to be able to know which cells exactly we're patching. So the idea behind two-photon targeted, it's very similar to single photon excitation. It's just that we're going to get a deeper penetration with two-photon excitation. Why? Well, in single photon excitation, you have one excitation photon that is going to excite your fluoropore enough to have it then emit fluorescence. And in two-photon excitation, instead of one, you're going to have two excitation photons. So you're going to have half of the energy needed from each of the photons that are going to be near simultaneously absorbed to have the same effect that the single photon would. So this has two consequences. So the first one is because it's half the energy, half the shift frequency, then the excitation wavelength is going to be longer. So this is going to mean lower scattering. And because we need the near simultaneous photon absorption, then that means that we're going to have a lower background signal because the chances of two photons being absorbed outside of the excitation range is going to be extremely low. So the basic consequence of all of this is that we have a deeper penetration, which can be quite useful depending on the type of cell that we want to go for. Now, what is the motivation? Why are we doing this? So as I said before, doing a patch clamp, even single patch clamp, is quite difficult, and doing multi-patch clamp is extremely difficult. Only a few laboratories in the world do it. And as I said, though, it's very useful for understanding the brain. So obviously, we want to automate this technique. And you may ask, has somebody done this before? Has somebody tried to automate it? Well, yes and no. So somebody has done it back in 2016, I think it was. But it was blind. So what does this mean? Basically, you're going to have the 4 pipettes going to the brain, and then just whatever 4 cells are found first, those are the cells that are patched. Now, obviously, this is useful because it can increase the throughput, but as you can imagine, it has a lot of disadvantages. So the first one is going to be that, obviously, the 4 pipettes are not going to be that close to each other because they could crash, and you don't know where, exactly where they are. So obviously, you're not going to be able to patch cells that are very close to each other. So that means that probably you're not going to see connections between the cells. And the second consequence is that, well, if you're going to patch, for example, in the cortex, you have a lot of pyramidal cells. So if you want to patch pyramidal cells, then fine, you can use the blind. But if, for example, you want to patch one of the interneurons that I was talking about, that they're not that common, they're less than 25% of the population, depending on the part of the brain, then obviously you're going to have an extremely reduced throughput with any blind approach. So you need to have this 2-photon multi-patching to be able to target these subtypes. Now, what does the robot look like? Well, the setup is actually quite similar to a patch clamp setup. So you're going to have 4 manipulators that are going to allow you to move the pipette into the brain. And inside each of the pipettes, you're going to have electrodes that are going to be connected to the amplifiers to basically pick up the signals from the patch clamp, and that's going to be relayed onto the computer. Then for the imaging part, you're going to have this here. So you're going to have a laser that is going to excite the fluorescent proteins inside the cells in the brain. And then the emission is going to be picked up by photomultiplier tubes. So really, the only thing that you need to automate this kind of setup is a pressure system that is going to apply the negative and positive pressure depending on the moment to achieve this seal and this break-in. And yeah, the pressure system is actually quite simple. It has a pneumatic and an electronic part, and everything is controlled from the software. So this is the software that we have designed, and I know it might seem like a lot of buttons, but trust me, it gets very little to get used to. We have done it as much user-friendly as we could. The idea is that for each of the 4 manipulator/4 channels of electrophysiology, you're going to have a distinctive color that is maintained through all of the GUI. And also the idea is that you're going to have different modules that are, each of them are functional. So this would be the imaging module. This is the manipulator control. This is the auto-patching section. Then this is the module for electrophysiology signals, basically the patch clamp signals. Pressure, and then we also have some controls here that are not needed all the time, so they're hidden. So you have, for example, heater control so that you keep the cells in a physiological range, and you also have cleaning controls so that basically you can reuse the pipettes and you don't need to change them after each trial, which is what it's technically done when it's what it's usually done when it's done manually, and then also some experimental notes. Now, also I would like to say that we have made it in such a way that if the experimenter has no idea how to do patch clamp, then the auto-patcher will do everything for the experimenter. But if you already know how to patch clamp, this could still be useful because it can, for example, avoid, you can have the cleaning part so that you don't need to change the pipettes, or you can also have all the pipettes be moved to the desired region that you want. So it really can help you no matter what stage you are in your patch clamp learning curve. Now, all of this work is based on this robot was already developed, or the beginning of the robot was already developed back in 2017, where it was used to do single in vivo patching. Now, in vivo patching basically is going to have the problem that the mouse is going to be breathing, and also there's cardiac movements, so the coordinates of your cell are not going to stay constant in time. So the automated part of the algorithm, which is basically this part here, starting with the cell approach, basically is that your manipulator is going to move in a little bit, then you're going to reacquire the target, then move a little bit, acquire the target, and so on and so forth, iterative process until the pipette is close enough to the cell to do the seal and the break-in. Now, for the Quadruple Xv Patching, which is the last thing we have developed, we actually went simpler because it's ex vivo, so it's basically brain slices, you're not going to have the movement that you had before. So the user only needs to acquire the pipette and the targets at the beginning to choose what cells the experimenter wants to patch, and then after that, everything is automated. Basically, the software will acquire a 3D stack, and through image processing is going to detect which is the plane where the cells are the most in focus, and those coordinates will be used for the pipettes to approach the cells directly in two single movements, in a diagonal fashion, by the way. And then once the pipettes are in contact with the cells, they will automatically, the software will automatically apply pressure, and the seal and the break-in will take place. And as I said before, we included pipette cleaning, which was first developed by Kolb et al. In 2016. And this will allow the experimenter to just reuse the pipettes over and over again. So then, again, the only thing the user does is just acquire the pipettes and the targets and sit back and relax. So this is an example of a quadruple patching that the robot did. So these 4 cells here, I hope you can see them. They have these targets. They are the ones that have been chosen. And I skipped the part where we take the 3D stack because it's not that interesting. So now here I'm changing to the IR. This is actually not necessary. I'm just doing it for purposes of you seeing now, hopefully, the pipettes are going to come in here, 1, 2, 3, and 4. And then as soon as they touch the cells, you can see here, it was already, the buttons for sealing were pressed automatically. And as you can see here, the positive pressure was released, and actually negative pressure is being applied where necessary. And if you see here the resistances, they are already achieving this gigaseal that we were talking about that we needed. So 3 out of the 4 channels were already getting there quite fast. And as you can see, the buttons therefore were unpressed because the software already noticed that it had achieved the gigaseal. And then blue took just a little bit longer, but it eventually got there. So the idea would be then this would be unpressed, and then the breaking buttons would be pressed. And so then at the end, we had the 4 cells broken into, and you can see here the resistances below 100, and actually all the membrane potentials were within physiological range of all 4 of them. And with this algorithm, we have gotten a seal yield of 54% and a breaking yield of almost 46%. And now just to finish, what does the future hold? Well, obviously quadruple in vivo patching is the next step that we're going into. And right now we don't know if that's going to include some iterative algorithm like the single in vivo patching, or if it will be more simple like the quadruple ex vivo patching, but that's something that we will see. And that was all, and thank you very much for your attention.
So thank you, Gemma, for the nice presentation. I am a little far on this topic, but I really find really interesting. There is any question on the room or perhaps on internet?
Hi, thank you so much for that wonderful presentation. I think it's a really interesting topic, especially when we get to the kind of temporal resolution you get. So regarding the last slide about the future where it's moving to in vivo, so that's really fascinating. So currently, what are the main challenges to overcome regarding adapting your current method for in vivo multi-patch clamping? And have you managed to get preliminary results from that?
No preliminary results so far. We do have like a slight idea in the sense of like we already had the in vivo single patching. I think obviously one of the difficulties is obviously the movements from the like breathing and the heart, obviously. But also the preparation of the craniotomy is actually quite essential to getting good recordings in vivo. So that's something that we will have to be, whoever does it has to be very properly trained, because the craniotomy quality is essential for good recordings. So yeah, I guess, yeah, there will be a lot of challenges, I'm sure. But yeah, those are the two main ones, I would say.
Thank you.
Any other question? So thank you, Gemma. Thank you for your presentation. And now we are going to move on the next presenter.
Yeah, so the next presenter is Professor Simon Schultz from Imperial College London. He's going to be talking about multiphoton imaging of 5 FAD mice performing spatial memory tasks. And I think he is live online, so...
Emma, can you hear me?
Yes. Yes, we
Good. I will just share my screen now, and hopefully this should work as well as it did when we tested it just before. Okay, so you can see my screen correctly? Yes. Good. So yes, I'm going to be talking about multiphoton imaging of 5x FAD mice performing spatial memory tasks. This is work done by a number of people in my group, particularly Marianne Go, Yimei Li, and Siegfried Prado have been the main contributors to what we're going to be talking about today. This is really sort of motivated by Alzheimer's disease, and just to give you a UK perspective on Alzheimer's disease. Dementia, of which Alzheimer's is a variety, is now the leading cause of death in the United Kingdom. It's the 7th leading cause of death worldwide. In fact, it took over as the leading cause of death only recently, only shortly before the COVID pandemic. It surrendered it, if you like, to the COVID pandemic for a short period, and it is now again the leading cause of death. It has greater economic impact than cancer and heart disease combined. A quarter of all hospital beds in the UK are occupied by people living with dementia. And the most common form of dementia is Alzheimer's disease. So we've, if you're looking for a problem in a medical problem to investigate, clearly Alzheimer's disease has got to be, one of the crucial ones to target. Now we're interested in brains and in the technology for investigating brains, and so the question to ask really is, well, what happens to brain circuitry during the progression of Alzheimer's disease? The areas damaged first are the hippocampus, which is a deep brain structure, which is crucially involved in particular types of memory, and also structures connected to it, like for instance the entorhinal cortex. And they show two particular pathologies: amyloid beta plaques and tau tangles. So the amyloid beta plaques are basically conglomerations of abnormal amyloid protein sort of clustered together that you find extracellularly. It may actually start in some cases intracellularly, but you find it extracellularly. And then you have these additionally, they have these tangles of tau protein, which are found intracellularly. So these are the two sort of molecular hallmarks of Alzheimer's disease. They aren't separate though, they're linked together in a complex biochemical cascade. It's not clear sort of, who the cause and who the effect is in this cascade. They both sort of relate to each other. Amyloid tends to occur earlier and tau later, but they are sort of linked together in this cascade, which eventually ends up in neuron death and impaired memory and cognition. Now, en route to that, amyloid beta in particular has been found to lead to aberrant excitability of excitable cells, neurons in the brain. So you both have aberrant excitatory activity, where basically you have hyperactivity, cells will fire too much. And of course, you've got this sort of complex network involving inhibitory interneurons coupled in with the excitatory neurons that are affected too. You have potentially compensatory inhibitory mechanisms. And in the end, you have network dysfunction, which causes the information processing operations that the brain does to be affected. So to investigate this, we need a model, and what we've used is this 5XFAD mouse model. It's got five, basically it's a mouse which has been genetically modified to have 5 Alzheimer's disease linked mutations. And they're in two sort of areas in the genome. There's basically amyloid precursor protein, and is introduced under a particular promoter called Thy1. There are these 3 mutations, the so-called Swedish mutation, the Florida mutation, and the London mutation. And then in addition, in PS1, there's also 2 additional mutations. What this does is it leads to high intraneuronal amyloid beta quite early on in the mouse's age. So mice obviously live only for a couple of years, so the timescales are much compressed with regard to humans. So there's high intraneuronal amyloid beta from 1.5 months, synaptic degeneration from 4 months, amyloid plaques forming from 6 months, and memory impairments found from 6 months of age. We use spatial memory in particular as a kind of a way to get at a more general memory impairments. So how do changes in neuronal circuits lead to cognitive impairment? That's one of the big questions we're asking here. And then how can we repair these changes? Okay, so we developed a platform technology for studying this. So one thing we want to be able to do is to image activity of populations of neurons in the affected areas, and particularly in the hippocampus, while a mouse is doing a behavioral task which lets us get at cognitive effects. So the way this works, so we're doing this in conjunction with 2-photon microscopy, as you heard in the previous talk. In this case, we use it to visualize the activity of neurons. We inject a virus into the hippocampus, which causes the neurons to express a genetically encoded calcium indicator, JGCAMP7. And you can see in this example here in the green cells here flashing away. As they fire action potentials, they flash away, and calcium is influxing into the cell, and then that causes an increase in the fluorescence that we can see. Now we do this, we can't image directly into the hippocampus because it's a little too deep, so we basically do an aspiration of a small amount of overlying cortex and implant an imaging window, which we can image through. Now, we have the mice doing behavioral tasks, so they're actually head fixed, so we have best access to the cortex. So they're head fixed in one place, as you can see here, but they run on this this basically carbon fiber chamber, which is floating on an airbed, and they can explore the chamber, do little maze tasks, and we can map out neuronal activity and how it relates to the to whether or not they can remember where they are in the task, for instance. So doing that, we can image activity of populations of neurons in area CA1 in the hippocampus during behavior. We can see an example here where we've been able to pull out. So the green channel here is the calcium signals. The red is another reference that's not activity dependent that we use. We pull out regions of interest corresponding to many hundreds of cells. And then we can see these sort of time series signals here. So what you're looking at here, the magenta line here is actually the position of a mouse running around a circular track. So it's repeatedly running, doing laps around this track. We, at the same time, record these time series from individual neurons. You can see the little calcium transients corresponding to action potentials being fired there. Then we detect those individual events and we give them basically a size here corresponding to the amplitude of the calcium transient. And so you've got this sort of rastergram there. We can then basically relate that to task properties. So here's an example where we've mapped out 5 so-called place cells. So the cells that respond to a particular location in place. And there's one on the left, you can see here, basically every time the mouse passes a particular location, we're placing a dot down there. So you can see it's selective for what particular location in space. These other cells are selective for different locations in space. We can then unroll that sort of down here across laps, and you can see that the location is pretty consistent from lap to lap. There's a little bit of drift, but it's fairly consistent there. Now this basically, it allows us to get it spatial memory. So if the animal remembers that it's been in this environment before, it sees particular cues, it recalls the spatial memory. So this is a good model for memory, for long-term memory for mice. And that's a crucial aspect of episodic memory, which is, I guess the thing we're really interested in and that Alzheimer patients sort of, are impaired in. So this slide shows results in a wild-type mouse. What does it look like in the 5XFAD mouse model of Alzheimer's disease? Well, we can see down on the left here a comparison between what we might see, the rasters we might see in a wild-type and 5XFAD mice. The 5XFAD mice are sibling controls, like of the sibling, like some will express the mutations and some will not. So we've got a natural control there, and you can see. It's not maybe particularly obvious, and just by looking at a handful of cells, the differences. Maybe there is a slightly broader tuning there. In fact, if you sort all of these place fields, so for each cell, we'll take an average down there and make that a row on this, and then show them in sorted order. And we do that for both types of mice. You can see. Basically, the background is higher in the 5XFAD mice. So what does this really mean? It means there is less information there. So there's a few things going on. The first thing you can see is that there is hyperactivity overall. So if you look at this histogram here, there is basically the red ones here, the red bars correspond to cells in the 5XFAD mice that systematically shift higher. There are more calcium transient events from the 5XFAD mice. The place fields, so basically if you measure the width of the area to which the cell's responding, they tend to be bigger in the 5XFAD mice. So the place cells are less selective, if you like, they fire to a larger area. You can measure this in terms of spatial information as a bit rate, a Shannon information bit rate, and in fact, the spatial information rates are lower in the 5XFAD mice. So we actually have a number that we can put for each, actually cell by cell, and basically what's the contribution of that cell to the memory in bits per second, and we find that those contribution in bits per second are decreased in the 5XFAD mouse. Now, we've then sort of gone on to do this in a forced alternation spatial working memory task. So it's basically the same, but rather than the mouse just running around in a circle, it has to, it goes down a central corridor And if it goes left on one trial, then the next trial it has to turn right, etc. It has to remember what it did last time. So what happens there? Well, what we find is even in young mice, the 5XFAD mice are impaired with respect to the control. So the controls managed to learn this task over 20 sessions, about 10 days. The Alzheimer model mice failed to learn it. Old mice, up 7 to 9 months of age, the wild types learn it. They're actually slightly slower than the younger mice, as you might expect, but the Alzheimer's are, again, sort of severely impaired. What's going on here? Well, one thing we found that this relates to is hyperactivity, so that actually the cells in the old Alzheimer mice have a tendency to be hyperactive, to be firing at higher firing rates, and less selective. So that's sort of what's giving rise to this. And then finally, so we're doing all the stuff I've told you so far. We've done in awake mice. We're imaging the brains of awake mice while they're doing a task. At the end of the experiment, we basically we kill the mice. We fuse the brains, and we basically we put them into this serial 2-photon tomography imaging system, which images 50 or 100 microns, sort of thick sections, tiles that across, slices it off. Images the next one, etc. And then after a few days, it takes a few days to do the imaging, we've got 5 terabytes of TIFF images in a directory and a lot of slices sort of on the bottom of the bath. So we can basically look at the whole brain in this way. And this is what you see here. So what we've done is we've injected methoxyXO4 so that we can basically label the amyloid plaques fluorescently, and we can then see where they are throughout the entire brain. We can see it's a little lopsided here. The reason for that is because we have this imaging window on one of the hemispheres that we were imaging in vivo, but we can then look at the other hemisphere and actually map out where, what fraction of area is occupied by amyloid plaques in each brain region. These are registered to the Allen mouse brain database, so we can then do statistics over mouse brains, across individuals. And when we do that, we find in this mouse model that the hippocampus and the thalamus in particular show most early, they show a sort of development of plaques. What we're now trying to do is to bring this back to the in vivo results and look at, whether we can predict the in vivo imaging results, the behavior from the specific pattern of activity in the ex vivo imaging data. So finally, to finish, we've validated a platform for characterizing the contribution of hippocampal neurons to cognitive tasks. We've tested it on this 5XFAD mouse model of Alzheimer's disease, finding hyperactivity, larger place fields, and lower spatial information rates. And then we've further applied that to a working memory task, and we see decreased performance in Alzheimer's mice compared to the wild-type siblings, alongside neuronal hyperactivity. So I will end it there, and thank you for your attention.
Thank you very much. Are there any questions in the room? Yes.
Hi, Dr. Schultz, thanks so much for your presentation. So actually, I'm from a lab in Singapore, and we are doing an experimental setup that is remarkably similar to yours in terms of the calcium imaging in mice and also a spatial memory task. So I think based on our preliminary results, we find quite similar results to yours. But the one thing that I guess has stumped us a little bit, and what we're also trying to work on, is trying to link what we know about the neurodegeneration to why the place fields widen as a result. So we'd like to hear your preliminary thoughts on just your working thoughts on how you might have drawn such a link.
Yeah, that's an interesting point, and that is, I guess, something that we're still getting at. I would say it in particular relates to the coupling of inhibitory interneurons into this sort of excitatory network. So both are affected. Effectively, you've got a situation where all of the cells could have wider wider tuning, selectivity to a wider area, but much of that is normally suppressed, so that effectively it's sort of sculpted out so that a lot of the selectivity, it is sort of narrowed down. And I think what's happening is we're having this disruption of the excitatory inhibitory network there that's disrupting that that sort of sculpting out by the inhibitory interneurons, and that's therefore broadening the spatial tuning. So that's sort of my hypothesis as to what's going on there. And I don't have data to back that up at the moment, but that's definitely something that we're looking at as well.
All right, thank you so much for sharing.
Thank you.
Any other questions? I think there are no questions online either.
Okay, so thank you, Professor Schultz, Simon Schultz. So let's move to the next presenter. The next presenter is not here, he's a pre-recording video. So I think it's more or less ready. The name of the presenter is Gali Restur Fardian Suandi from Indonesia. And he will talk about the spectroencephalogram, spectral analysis on virtual reality sickness from screened and head-mounted display. So I think it's ready.
Hello, good afternoon. First of all, I would like to introduce myself. My name is Gali Restu Fardian Suandi. I am a student from Department of Physics, Institute Teknologi Bandung, Indonesia. Today, I would like to talk about a part of my research entitled Easy Spectral Analysis on Virtual Reality Signals from Screens and Head-Mounted Display. Okay, I'll divide my talk into 4 parts. The first part, I will tell the background and motivation of this research. Secondly, how this research was carried out will be explained. Third, I will try to discuss the result obtained, and I will summarize my presentation at the end. Okay, the virtual reality or VR technology had advanced significantly in recent years. Many new devices have been introduced to create games, movies, and other immersive experiences, suggesting that they are on their way to become a mass market products. However, VR sickness may occur when a person immerses into the VR environment. VR sickness is a motion sickness-like disorder, often occurring to a person exposed to an environment where the visual and proprioceptive motions are conflicting. A person with VR sickness suffers from headaches, stomach awareness, nausea, disorientation, sweating, fatigue, and even vomiting, which raises safety and health concerns for current VR platforms. Therefore, VR sickness is considered a major hurdle for the wide acceptance of VR application. To investigate any VR reduction methods, it is necessary to have tools to evaluate VR signals efficiently and also effectively. The Simulation Sickness Questionnaire, or SSQ, has been widely used to measure the amount of VR sickness experienced during VR exposure. However, the subjective evaluation method has some disadvantages. It is usually performed before and after a VR signals experiment, and due to the large length of the questionnaire, it cannot be done in real time, hence cannot describe the changes of VR sickness during the exposure. As a result, it is difficult to detect the emergence of VR sickness or get the details of VR sickness development by using this method. To overcome the limitation of subjective VR sickness measure, objective VR sickness evaluation methods based on various physiological signals, such as electrocardiography, pulse rate, blood pressure, ECG, salivary cortisol level, EOG, and EEG were tested. Such physiological signals can be measured continuously and precisely. Different physiological signals are associated with different VR signals theories. For example, the EEG signal is usually related to the sensory conflict theory. The sensory conflict theory is that situation that provoke VR signals can be characterized by a condition of sensory rearrangement, in which the motion signals transmitted by the visual and visible system, or maybe other proprioceptive system, are mismatched with one another, or different from what is expected from previous experience. Many researchers measure EEG and motion sickness studies based on sensory conflict theory. The test if the EEG measured can indicate the level of motion sickness objectively. So in this research, I'll try to find the link between the SSQ and the EEG-based evaluation of VR sickness. Okay, the EEG data acquisition was carried out with 20 volunteers, male, 20 to 25 years old, and all volunteers did not have a history of serious illness, neurological health problems, and were not taking drugs. On the day of measurement, all subjects were ensured to be in a state of sufficient sleep and meet the health requirements according to the COVID-19 health protocol. All volunteers needed to be inexperienced in VR simulation, meaning they have not experienced VR technology for more than 60 minutes. We applied a multi-stage experiment. After applying the EEG cap, the VR device as the HMD, and the screen was attached, and the volunteers entered the VR environment. There are 4 different videos that are given. First, videos of static views. The second, videos of color changes. The third, videos that move in one dimension, forwards and backwards. And the last is a moving video that is more complex. I choose the roller coaster videos. The recorded data is processed through 2 steps to obtaining the desired information. The data from the EEG recording enters the signal preprocessing stage. This preprocessing aims to eliminate noise and artifacts that appear during recording. The EEG devices generate offset data values. Therefore, the first step to do this is data centering. The centering stage is carried out by the deviation process using the average reference method. All data are averaged, then each data is subtracted by the average value. A processing technique on the central signal was performed to get the clean EEG signal. Signals in the delta, theta, alpha, and beta wave range are obtained through a process using bandpass filter. The filtering process resulted in the EEG signal in the many wave range only without unwanted noise, such as the AC frequency rate 50 or 60 Hz and blink mostly below 0.5 Hz. In the last stage, filter extraction is carried out using power spectral density calculation using the Wells periodogram. The PSD value analysis was conducted by calculating the average value of PSD changes by using the initial eye-opening condition as the baseline. Then the value of the change in PSD is averaged for a subject, and the result are plotted in alpha, beta, delta, and theta waves. Okay, the SSQ results show a significant increase in the value of some symptom categories. The volunteers in general feel stronger symptoms than before as the video changes. As we can see in the graph, on the change from resting state to the second video, there was no significant increases in symptoms except in the oculomotor category. However, after the third and fourth videos were given, a significant increase in symptoms occurred in almost all categories. nausea, and oculomotor. Also, in the total score, the increase is visible significant. Based on various studies that have been carried out, the symptoms of nausea and oculomotor are related to the frontal, parietal, and central area of the brain. Okay, after obtaining the power spectral density, it can be seen that there is a difference between the stimuli in the form of VR through the screen and HMD. The blue color indicates a change in the PSD throughout the video, while the green color indicates an insignificant changes. A significant change from PSD, mostly when volunteers were given VR through HMD. Notable differences appear in the beta low and the beta high frequency ranges. In general, it can be seen that the frontal area is almost always activated in the alpha range frequency, both in the case of screen and HMD. However, the results cannot be directly related to SSQ, as well as the results of research that has been carried out by various researchers. Therefore, some adjustments are needed in the protocol as well as the physical quantities to be analyzed. Okay, so that's a little sharing of the result of my research. If you have questions, suggestions, or input, you can send them via the following email. I am very happy to receive it. Thank you very much.
So there is no question for this presentation, but I invite you to follow the discussion online in the application of the Congress, or also writing him to his email. So next presenter comes from the University of Valladolid in Spain. His name is Eduardo Santamaria, and he's going to talk about a novel platform for implementing novel brain-computer interfaces. So Eduardo, your turn.
Okay, now. So thank you, Javier. Today I'm presenting Medusa, which is our novel platform for building modern and non-invasive brain-computer interfaces that we have developed in our lab. And first I'm going to make a brief introduction about brain-computer interfaces. These are systems that allow real-time communication between a human and an electronic device using just the neural signals of the user. And they have this architecture, which is, we use typically the EEG signal, the electroencephalography, which measures the electrical activity of the brain. So the user usually has an EEG headset, which is sending in real time the signal to a software, which has typically a signal acquisition module. This EEG is processed in real time using different techniques, signal processing, preprocessing, feature extraction, feature classification with machine learning. And afterwards, this EEG is translated into commands that reflect the intentions of the user, and they are sent to an application that executes the commands and offers feedback to the user. So these three modules, the signal acquisition, signal processing, and application, is what we call a Pci Platform. So as you can imagine, this kind of software is a little bit complex to develop. It has a very specific modules and functions, and currently we have very few solutions for BCI as BCI platforms. Mainly we have BCI 2000 and Openvive. BCI 2000 is the oldest one. It was developed in 2000, around that year. And it only has very few functions. It has P300, base applications, motor imagery. And when you control it, it feels like a little bit old. Afterwards, it came Openvive. So, okay, it's not the last version of the presentation, but it's fine. Afterwards, we had Openvive, which is a more modern platform, which implements more functions such as neurofeedback and steady-state visual evoked potentials. And an important feature is that they are both programmed in C++, and I'll come back to this later. And we have other platforms that are not very used and are discontinued for several years, like BBCI, BCI++, XBCI of BF++. I'm not going to talk about them. And all of these platforms, this landscape of BCI platforms, leaves several limitations for BCI research, and especially on other neuroscience experiments, because they have very complex architectures with highly coupled models, which is not useful for if you want to develop new applications, new features, or change some of the functionalities of the platforms. They are quite complex, and the code is in C++, which is a very low-level programming language, which makes it difficult to develop new features. And the current platforms especially are not designed to implement your own experiments. They come with a predefined set of functions. Especially Openvive, it allows some modifications and the development of some modules, but it's quite difficult to do it. So in our lab, we wanted to overcome these limitations, and we set up the path to build a new platform to design, develop, and test new BCI paradigms and experiments. So we developed Medusa. And this platform is specifically developed for research. It's developed in Python, a high-level and multi-platform programming language, which is very useful to develop new features and especially quick prototyping of new modules. It has very autonomous components connected by very simple communication protocols, so you can develop new modules and new functions very quickly. It's flexible and it's very scalable. Okay, so these are the main highlights of our platform, and especially it can be divided into two key components, which are the Medusa kernel and Medusa Platform. Is the two modules that we have developed. So, well, I'm going to pass this, and I'm going to go to the main functions of this Python package. This Medusa kernel is a signal processing toolbox for biosignal processing. It is indexed in the Python package index, so you can install it just like any other Python package with pip install Medusa kernel. And it has a quite complete set of functions. It has low-level functions that don't assume which signal we are processing. It has preprocessing functions, transforms, machine learning, local active geometrics, connectivity. Okay, so it's quite complete. And afterwards, we have a few high-level functions, especially developed for BCI, which are especially BCI models for P300 speller, steady-state visual potentials, code-modulated visual evoked potentials, motor imagery. I'm going to talk about these BCI paradigms later, very briefly. And also we have a module for neurofeedback, okay? And of course, we take advantage of the wide range of libraries that we have available in Python, and this simplifies the development of the platform. So this is also important. Okay, this is the other component. The other key component of the environment is Medusa Platform, which manages a desktop program which manages the other functions that we saw in the architecture. We have a signal acquisition module which is based on live streaming layer, which is a pretty standard protocol nowadays, especially for research, to acquire biomedical signals. And this protocol detaches the functions of signal acquisition from the specific equipment that we are using. So Medusa is it's very compatible with most commercial and even non-commercial equipments for signal acquisition. We also have real-time charts for visualizing in real time the signal, especially now currently they are implemented time and spectral representations. Afterwards, I'm going to show you the main window of the application. And we also have what we call the applications, which are the central part of Medusa Platform, which are programs that are executed within the platform using its functions, but are independent. So these little programs are useful to perform several experiments, BCI experiments or neuroscience experiments. It's very flexible, the platform. You can develop and share your own applications in the app market of our website, which is very useful. And currently we have 4 BCI applications for an ERP speller or P300 speller, a code modulated visual potential-based speller, motor imagery, and neurofeedback. These are the 4 functions that are currently implemented. And we have other applications, a simple recorder, some working memory task, and you can record the signals at the same time that you are doing the tasks. And we also have some demonstrations for neuroscience courses, such as P300 and visual evoked potentials demonstrations. So this is the main window of the platform. We've employed a lot of time to make it also visual and easy to use. So I'm going to explain just the main parts of the window. Here we have the button to configure the input data streams, the live streaming layer configuration. You can configure as many input streams as you want. So you can have 2, 3, 4 EEGs at the same time, or other biosignals, ECGs, whatever you want, which is very useful in a lot of applications. Then we have the apps panel, the applications panel, where we have the applications that have been installed in the platform. You can launch here the applications, configure them, etc. Then we have a log panel to display important messages. And afterwards, we have the real-time charts. This panel is fully configurable. You can display as many charts as you want on demand, and you can attach them to different input streams. You can do whatever you want. So this was the main window, and now I'm going to make a very brief presentation of the BCI applications that are currently implemented in the platform. This is a classical P300 speller. If you are familiarized with BCIs, this was the first BCI speller that was developed. We also have a code-modulated visual evoked potential speller, which is nowadays one of the fastest BCIs for communication and control of devices. This is one of the more researched paradigms that are currently, it attracts a lot of attention from the research community nowadays, and it's fully implemented. It's made with Unity because it requires a very precise time synchronization between EEG signals and visual stimuli, because the stimulus are very quick here, and you can do it also with Unity within the platform, which is very useful for this kind of applications. We also have the motor imagery application. It's a very simple application to record motor imagery signals. And finally, we have the neurofeedback module, the neurofeedback applications with different functions and signal processing algorithms. You can configure and choose them in a very easy way. So in the discussion, I want to make a very brief comparison between Medusa and the other 2 main platforms that are BCI 2000 and Openvive. So the first thing that we have to keep in mind is that Medusa is developed in Python, which is a high-level language, programming language, which simplifies the whole chain of developing the platform first and also the applications, which is very useful, I think, in a research environment because you can use NumPy, scikit-learn, TensorFlow. You can implement deep learning models within the platform and use them online, which is pretty useful nowadays in BCI. The other thing is that Medusa is under heavy development now, so I just presented the functions that are currently implemented, but more functions are coming soon. We are developing new applications, new charts for the real-time charts panel, and we also have the offline analysis with Medusa kernel, which is not available in the other two platforms, and this is very useful to replicate the results of online and offline experiments, which is also very useful. And obviously, you can design and share in a very easy manner your own applications and experiments. This is also a feature that is quite difficult in BCI 2000 and Openvive, which are the other two main platforms that are mainly available today. So just to conclude my presentation, Medusa is a new environment that we have developed to build developed and test new BCI experiments and neuroscience experiments. It's specifically designed for research, so it's very flexible and compatible with a lot of devices. You can implement several. Nowadays, it has implemented several BCI applications, as you can see, on other neuroscience experiments. But you can develop your own experiments. More features are coming soon, so please check the website and our Twitter to stay updated on the platform. And I encourage you to visit our website. You have to keep in mind that we are still building it, so not all the web pages are currently available, but it's functional, and you can download the application, see the app market, download your applications. Get the source code, etc. So you can visit the website too. We'll finish it in the coming weeks. Okay. So that was my presentation. Thank you for your attention. And if you have any questions, I'll be happy to answer.
Thank you, Eduardo. I'm sure it will be a very nice and helpful application platform for researchers in the near future. So there is any question in the room or online?
Hi, thank you so much for your presentation. So my question is regarding the use of Python rather than the traditional C++. So given that Python is quite a high-level language, do you find that there is significant enough real-time latency, because Python is so much slower than C++, that it causes some disparity when you compare it to the results you get from C++, especially for the online analysis?
That's a very interesting point. The thing with Python is that you can call C or C++ libraries also, and they are very quick. So you can implement very specific functions on these languages, which as you said, they have higher performance in terms of speed and computational power. But these modules, these functions are pretty specific. So you can develop the main platform, the graphic user interface, and these kind of things in Python, which simplifies the whole chain of development. And you can then implement your specific functions, your key parts in C or C++. So I think what Python makes here is to broaden the compatibility. You can use a lot of functions that are developed within the research community right now, deep learning, machine learning. You can stay updated in all the signal processing, which is also very important. And these kind of things with C++ are very difficult, I think, because I think you would have to develop everything on your own, which is why the other platforms are not updated and are not currently used, because they are outdated in the end. So thank you for your question. It was very interesting.
Any other question, or in that case, we can move to...
So the next presenter is my co-chair, Javier Gomez-Pilar. He's going to talk about overnight slow waves differ in children suffering from sleep apnea and might characterize their cognitive impairment.
So thank you. Thank you, Gemma, for the presentation. Now it's done. So this study is a collaboration from the University of Valladolid and also the University of Missouri. And here I will try to explain the capability of the slow waves during sleep in a sleep apnea patient, pediatric sleep apnea patient, in order to predict the cognitive impairment that several times they have. So first of all, let me explain what is a sleep apnea disease. This is a disease, a very highly prevalent disease that affects both to adults and also children, up to 6% of the population in children. And during the night, these patients suffer from total occlusion, that's called apnea, or partial occlusion, that calls hypopnea, of the upper airflow. So the gold standard for the diagnosis is the PSG, that is the polysomnography. And the polysomnography consists of different signals. One of them is the EEG, the electroencephalogram. And also the treatment, regarding the treatment, the typical treatment is to remove the adenoids and the tonsils, but under certain conditions. The first condition they must meet is apnea-hypopnea index higher than 1 to 5 mm per hour. But also they must have other conditions like sleepiness, behavioral problems, learning difficulties, among others. The problem here is in the clinical practice, most of the times the physician don't evaluate these behavioral problems or cognitive problems. So in this context, we propose to evaluate these cognitive impairments by using the EEG recordings from the polysomnography. This is based on previous findings in which we find first that there are some alterations in low frequency bands, specifically in delta, between 0.1 and 4 Hz. And this alteration is related with impairment of the verbal function. We also see in the literature, in the previous literature, that the regularity of the EEG signal in delta 1, that is between 0.1 and 2 Hz, indexed by the spectral entropy, is strongly related with also this verbal function. And finally, in a previous study of us, we found that sleep apnea patients suffer from an increase in the powerful of the spectrum in the very low frequency band. So we ask if these two issues, these two conditions are related between them. So in this context, The objective of the study is to evaluate the potential abnormalities in the slow-wave EEG activity, electroencephalogram activities, between children suffering from sleep apnea and healthy controls to analyze its relationship and to try to predict the cognitive impairment in these patients. So for that purpose, we recorded several polysomnography, extract the EEG from that polysomnography, and compute different spectral parameters, and finally perform a correlation analysis in order to see the correlation, the association between these features of the EEG and different cognitive and behavioral functions. So first of all, we need to record the polysomnography, and we divided them in 3 different groups: no OSA as a control group, mild OSA, and moderate to severe OSA, that is obstructive sleep apnea patients. So as you can see in the table, there are no differences between groups in age or sex, but there are high differences in the apnea-hypopnea index. That is normal because it's a condition to differentiate between those groups. So first, as I said, we recorded the polysomnography of all these children, and from the polysomnography we can extract several clinical and clinical features like apnea-hypopnea index, respiratory arousals, spontaneous arousals, among others. And also the EEG, that we recorded the EEG in 8 different electrodes in the scalp and recorded under a sampling rate of 200 Hz. And finally, all the children performed 6 different cognitive and behavioral tests in order to assess its capabilities. So, well, after the acquisition, we perform a preprocessing step divided in 4 different steps. First, the re-referencing to the average activity of all the electrodes. Then the typical filtering between 0.1 Hz and 70 Hz. But afterwards, we are going to filter that only to see to the delta 1 band. And we will also remove the line artifact by using a notch filter. After that, an automatic rejection of the noised epochs, and we also remove the first and the last epoch of the overnight recording in order to eliminate those epochs in which the patient are not really sleeping, they are awake. After that, we perform a spectral analysis using a Welch transform and compute 3 different parameters. The first one is the peak power in delta 1 band. The second is the frequency of that peak. And lastly, the spectral entropy in delta 1 band. After that, we perform a statistical analysis in 2 steps. First, a comparison analysis, a between-group comparison analysis using the most appropriate test in each case, Kruskal-Wallis or Marron-Whitney, and also a correlation analysis within each group in order to see the correlation between cognitive and spectral EEG parameters. We also perform a bootstrap approach in order to minimize the differences in the imbalanced population. And this is the summary of the main results we found. Well, regarding the between-group comparison, we can see here in horizontal are the 3 different parameters, so spectral entropy, peak frequency, and frequency in the slow wave band, in delta 1 band. And in vertical, the control group, mild sleep apnea group, and moderate to severe apnea group. So main findings, as you can see, is these differences in which we compare group 1 versus group 3, that is control group versus moderate to severe group, in peak frequency and the frequency of that peak. And we see several differences in with spread in the scalp for these 2 parameters. So This means that these two parameters, peak of the frequency of that peak in delta 1, could be a potential biomarkers for sleep apnea identification. So regarding the correlation analysis within each group, we see that there is no differences in control, no differences in mild sleep apnea patient, but very high statistical differences, correlations in the severe, moderate to severe sleep apnea group. So this means that in horizontal there are the different spectral EEG parameters for all the electrodes, and in vertical are the different tests, cognitive and behavioral tests, and also clinical variables we have for these patients. So what can we see here is first that spectral entropy and the peak of the frequency in delta 1 band is strongly correlated here for moderate to severe sleep apnea patient, meaning that these two parameters could be a potential biomarkers for predict the verbal impairment in severe sleep apnea patients. Another important finding is the correlation that we see here between spectral entropy and the peak of the frequency in delta 1 with sleep apnea index, apnea-hypopnea index, and respiratory arousals. This means that these two parameters could be also potential features for automatic sleep apnea identification. And finally, other important, remarkable finding is that there is a correlation between the irregularity of the delta 1 band of the EEG with SPS, that is a marker for sleep homeostasis. This accounts for that irregularities of slow oscillation could be a marker for sleep disruption. So lastly, to summarize the main findings, we saw that first, the three spectral measures we calculate could be potential biomarkers for sleep apnea identification. We also see that at least two of these three parameters, the peak of the frequency of the delta 1 and the irregularity indexed by spectral entropy in delta 1, could be potential biomarkers for cognitive impairment in sleep apnea patients. And finally, that the irregularity in delta 1 band indexed with spectral entropy could be a good estimator for sleep apnea fragmentation in sleep apnea patients. So just to finalize, some limitations of the studies. First, as you see, is the imbalanced population, mainly for the moderate to severe group, but however, we tried to minimize this effect using a bootstrap procedure. The second limitation is the very specific range of age of the patient, from 5 to 5 years old. We try to minimize this effect using the age as covariable of the partial correlation we performed. And finally, that we don't see any significant differences for mild sleep apnea group, but this is probably due to they are very close to the threshold in which the mild and control group is divided. So in future studies, we will try to recruit a subject in other ranges of sleep apnea index. So that's all from my side. This is the co-authors of the study that I want to thank all of them, especially to the first author, Gonzalo César Gutiérrez Tubal, who is not here today, but I want to thank all of them. So thank you.
Thank you very much. Are there any questions in the room?
So good morning. I'm Professor Steens from VUB Brussels, and I would like to know if there is a plan for other correlation studies. So now you have only EEG, but there are many more parameters that you can study in sleep polysomnography. Are there any plans?
Yeah, so we are planning the continuity of this research using more complex analysis, in particular correlation analysis using a network, a network of correlational networks it's called. So it's a technique that is commonly used in other fields, but we think it could be very useful here. And yeah, we are planning to do so, yeah, in future studies.
And then if I can have a second question, just from curiosity. So there are observations, the shift in the frequency and higher peaks. Mm-hmm. How does that physiologically, neurologically correlate with the problem? Very hard question, probably.
This is very interesting question. This is why we work with physicians, because actually they must to answer this kind of question, of course. I am an engineer and I don't know how to why is the underpinnings of such peak. Perhaps we thought that it's related with arousals. Because you know, when sleep apnea patients have arousals during sleep, and this could be change the spectral power in very low frequency bands, especially in delta 1. There are other studies that inform of this. So we think that it could be related with that, but of course we need further analysis to ensure this question.
Okay, thank you.
Thank you.
Any other questions? Okay, and I think we'll move on to the next presenter.
Yeah. Okay. Hola. Hello. The last presenter is Víctor Rodríguez, who comes also from the University of Valladolid. And he will talk about a particular assessment of the influence of the line artifact in Imagery, magnetoencephalography, and EEG, electroencephalography recording. So, Victor, when you want.
Thank you very much. Thank you to the audience for attending this presentation. In this study, I'm going to present how the power line interference, the power line artifact, influences the functional connectivity analysis. This is an almost ubiquitous artifact, but there's little known about how it influences widely used analysis that are functional connectivity analysis. First of all, I'm going to use recordings from two neurophysiological techniques, electroencephalography, EEG, and magnetoencephalography, MEG. These techniques are able to record neural activity with a very high temporal resolution and in a non-invasively way. We can analyze the signals in several ways. By analyzing the individual time courses of each sensor or source from a network graph point of view, or analyzing the pairwise interaction between each pair of sensors. The functional connectivity, that is called this field that analyzes the individual interaction between pair of sensors, between pair of brain regions, is a field that aroused the interest of scientific communities like a couple of years ago. Nonetheless, there is still a flourishing topic that, with an increasing number of published articles each year. These functional connectivity metrics is used to analyze not in the broadband, but in specific frequency ranges. These frequency ranges are as you can imagine, the classical frequency bands, delta, theta, alpha, beta, and gamma bands. Okay? In gamma band, ranges from 30 to 70 hertz, and that it contains the frequencies where the power line interference is present. That is common to be around 50 or 60 hertz. That it is reasonable to consider whether this power line interference that comes from the power line affects functional connectivity performed in that band, in gamma band. Okay. And then the two research questions of this article arose. The first one is related with the power line interference itself. Do this interference influence functional connectivity analysis? And the second one is related with the filtering process that is usually performed to remove this interference. Do this filtering process affect the functional connectivity analysis? To answer these questions, we have used 20 MEEG recordings. 10 of them were EEG recordings with a sample frequency of 500 Hertz and 32 sensors, and the other 10 recordings were MEG recordings with a sampling frequency of 1000 Hertz and 160 channels. These signals were projected to the source space by means of the S-Loretta algorithm, and then the sources were grouped in regions of interest by using the Desikan-Kiliani atlas. Then the source level signals were filtered between, bandpass filtering filtered between 1 and 70 Hertz to limit the bandwidth of the noise. And then we apply a notch filter with different characteristics to assess how these characteristics affect the influence of the power line in our results. We vary the filter bandwidth, the filter order, filtering window, and the sampling frequency of the signals. Then we use these signals, and we not filter these signals. We narrow band filter these signals using 1 Hz filter bank, thus having a pseudo-continuous frequential evolution of the connectivity. That is, we have a connectivity matrix for each specific frequency. This way of analyzing the functional connectivity is called high-frequency resolution networks, and you can find more details in the paper below. In this way, we will be able to assess, to analyze, to visualize how the power line interference affect each step of each frequency of the signal. Okay. Let's analyze the results. First of all, we analyze it whether the power line interference itself and not its associated filtering is affecting the connectivity. To do that, we plotted the strength, that is the average connectivity value, for different sampling frequencies, 250, 500, 750, and 1000, that is the original frequency. It can be appreciated that around 50 Hertz there is the power line interference, and it can also be appreciated that the sampling frequency has a little impact in the results. This is the result for the MEG database, but for the EEG database, similar results can be observed, maybe in a lesser extent. Furthermore, we also analyze the influence of the notch filter bandwidth. It can be observed that if the filter is wide enough, a similar shape of the artifact appears, two peaks joined by the low strength levels. Nonetheless, if the filter is not wide enough, that for example for this 2 Hz wide filter, 2 Hz width filter, the artifact yields lower amplitude. This is for the MEG database, and for the EEG database, a similar pattern can be appreciated, this time yielding higher amplitude for the narrow filter. Furthermore, we analyzed the influence of the filter order. We tested for values of it, 500, 1000, 2500, and 5000. It can be observed that higher filter order yields higher amplitude artifact. The same can be observed for the EEG database, this time yielding higher amplitude artifact again. Besides, we analyzed the influence of the filtering window. It can be observed that this parameter barely influences the results, neither for the MEG database nor for the EEG database. And finally, we assessed if the sampling frequency of the signals influence the results now that we have filtering applied. It can be observed that sampling frequency of, with sampling frequency of 250, 500, 750, and 1000, the sampling frequency affects the artifact with higher sampling frequencies, yielding lower amplitude artifact, both for the MEG and for the EEG databases. Three main conclusions can be drawn from this study. The first one is that although the power line interferences, the interference and its filtering process affect the functional connectivity analysis, it affects differently MEG and EEG databases. Next, we have concluded that the power line artifact, the power line interference affects itself the functional connectivity analysis, having the sampling frequency little impact in the results. Finally, we have also observed that the filtering process associated to the functional connectivity also affects with the notch artifact, with the power line interference, also affects functional connectivity, having the filter bandwidth, the filter order, and the sampling frequency of the signals a great impact in this interference. This is a preliminary study, and we conducted it with a limited sample size. We also used a sparse value of the parameters assessed. Nonetheless, we think that we have yielded some interesting results, and it worth continue working in the study. Because one main conclusion can be drawn, and this is that the functional connectivity analysis performed in gamma band should be very carefully interpreted. Now we are going to continue this study, filling the gaps obtained in the parameters sets, and we are going to study also other forms of removing the power line interference, such as adaptive filtering or source separation techniques that may also be capable of removing, at least partially, the influence of the power line interference in the functional connectivity results. And that's all from my side. Thank you very much. I would love to hear your questions and comments. Thank you.
So thank you, Victor, for the nice presentation. There is any question in the room or online?
Same author. So thank you for the nice presentation. So you do a multi-parametric study on the filters, the properties of the filters and so on. But I could not extract from your presentation what are the default parameters, because you can scan the bandwidth of your filter, the order of your filter, but what are the default parameters?
Okay, this is a really interesting point. I use 1000 as filter order, 2 Hertz as filter bandwidth, Hamming window, and the default sampling frequency of each database, that is for EEG 500 Hertz and for MEG 1000 Hertz. It's very interesting to perform a multifactorial analysis, but these results are really very high, computationally expensive. So we are now preparing like a computational cluster in our lab, and we are going to repeat the analysis in a multifactorial way.
Yes, and then you hope to find the optimum combination.
Yeah,
okay.
Of course.
Okay, thank you.
Any other question? So this is the last presenter, so I think we can conclude this session. Thank you everybody to listen, and the opportunity of the organizers to be here. So thank you very much, everybody.